Remember when your biology teacher told you stuff moved from here to there inside the cell, but never actually said how? Well, in reality, there are these tiny motor proteins that walk on tracks called cytoskeletons, carrying things around. They're kind of like ants moving food inside their nest. The cytoskeleton is a network of filaments that covers the whole cell. It's a cellular structure that helps the cell maintain its shape and internal organization. It provides mechanical support, enabling cells to carry out essential functions like cell division, anchorage, and movement. There are three types of cytoskeletons, the thinnest one being microfilaments, then intermediate filaments, and finally the thickest, microtubules. There are also three main families of motor proteins, dynein, kinesin, and myosin, and they all convert chemical energy from ATP to mechanical energy as they move. Both dynein and kinesin walk on microtubules, and they do so in a kind of hand-to-hand way, never letting go of the track, because they often have to carry some heavy cargo. The polarity of microtubules also affects the motor's direction. Dynein goes from a negative end to the positive one, which means it takes things from the cell membrane towards the nucleus, whereas kinesin goes the other way around. They both share similar functions, taking things from the cell body to nerve endings and vice versa, transport of organelles between the Golgi apparatus and the plasma membrane, and the transport of mRNA. Kinesin is a bi-headed molecule, and when it walks, one head binds to the microtubule and an ADP molecule is released. This allows for an ATP molecule to bind in its place. The binding of this ATP causes the mechanical element to zipper up along the head's core, displacing the partner head from a rear side to a forward side. The first head then hydrolyzes the ATP molecule into ADP and a phosphate. When it releases the phosphate, it loses its affinity for tubulin, so it releases from the track. While this happens, the second head, which was moved forward, is binding to the microtubule and releasing an ADP molecule to then bind to an ATP in its place. That's how kinesin takes one step, and then the cycle starts all over again. Myosin works on another type of cytoskeleton, actin microfilaments. However, its molecular structure is pretty similar to that of kinesin, and it too has two heads. Myosin, though, does not walk in the same way. In muscle myosin, for example, when its actin binding domain binds to the actin filament, it causes it to release a phosphate, causing a large forward rotation of the mechanical element of the head. The core will then release an ADP molecule and an ATP molecule will bind to it, causing it to dissociate from the actin filament. The core hydrolyzes the ATP into ADP and a phosphate, and it recogs to start the cycle again. In muscle, myosin is placed in this way, so that as it walks, it pulls the filament closer together, resulting in a contraction of the muscle. Myosin is many times responsible for the movements of cells and for cell division, just like kinesin. So the next time you study movements in cells, remember it's our ant-like protein friends doing all the hard work.